Here's the script. Picture this: along a muddy waterhole in Australia's Northern Territory, an army of ant swarms towards something that should be poison to touch. Within seconds, dozens of them are locked onto a creature nearly bigger than themselves, dragging it down, and within minutes, it's gone. What they've just killed is one of the most feared invaders in Australian history, a toad so toxic it can kill a crocodile. And the strange part is, scientists didn't plan this. They found it. To understand why this moment matters, you have to go back to 1935, when Australian sugarcane farmers were desperate. Beetles were destroying their crops, and someone had the idea to import a toad from South and Central America that supposedly loved eating beetles. So more than 100 cane toads were released into Queensland with the hope that they'd solve the problem quietly and disappear into the background of farm life. Instead, the beetles kept munching away at the crops largely untouched, because it turned out the toads couldn't even reach the beetles living high up on the cane stalks. But the toads themselves had no trouble surviving. They ate almost anything that fit in their mouths, they bred at an incredible rate, and they carried a defense that no native Australian predator was prepared for. That defense is a milky toxin stored in glands on their backs and behind their eyes. It's powerful enough to stop a heart, and for generations, Australian predators had never encountered anything like it. Snakes, goannas, quolls, even large birds would try to eat a cane toad the way they'd eat any other frog, and within minutes many of them would be dead. This is the part of the story that made cane toads so devastating. It wasn't just that they spread, it's that their presence quietly wiped out the very animals that might have eventually kept them in check. Populations of northern quolls collapsed in parts of the Northern Territory. Some monitor lizard populations dropped by huge margins in areas where toads moved through. The toad didn't need speed or strength. It just needed predators dumb enough, or hungry enough, to try eating it once. Today, estimates put the cane toad population in Australia at more than 200 million, spread across over 1 million square kilometers of the country's north. That is a staggering scale for an animal that arrived from another continent less than 100 years ago. A single female can lay tens of thousands of eggs in one sitting, and she can do this more than once a year. Multiply that across millions of breeding adults, and you get an avalanche of new toads entering waterways every single wet season, far outpacing anything that hand collection or community toadmusters have ever been able to remove. People have tried. Volunteers across Queensland and the Northern Territory have spent decades on organized toad collection nights, and while it helps in isolated pockets, it barely dents the overall population, because for every toad picked up, thousands of eggs are still hatching somewhere else. For a long time, that felt like the end of the story. An unstoppable, poisonous invader with no real predator anywhere in its adopted home. But researchers at the University of Sydney, led by biologist Rick Shine and his research group known as Team Bufo, started paying close attention to what happens to cane toads in the days right after they leave the water. That short window turns out to be the toad's only real moment of weakness, and it's the reason ants have entered this story at all. When a cane toad tadpole finishes developing, it transforms into a tiny toadlet, sometimes barely the size of a fingernail. At this stage it hasn't grown into the tough, poison-soaked adult it will eventually become. Its legs are weak, its jumps are short, and worst of all, its body loses moisture quickly, which forces it to stay close to the damp edges of ponds and waterholes instead of spreading out across dry land. That single limitation creates a bottleneck. Hundreds of these baby toads end up crowded along the same narrow strip of muddy shoreline at the same time, all trying to survive in an environment that offers them very little cover. And along those same shorelines live Australia's meat ants, an aggressive native species that patrols the ground constantly in search of anything it can drag back to the colony. Shine's team discovered something remarkable when they compared how native frogs and baby cane toads reacted to these ants. Native frog species have spent millions of years alongside meat ants and instinctively avoid them, hopping away the instant one gets close. Cane toad metamorphs never developed that instinct, because in their original South American habitat, they never had to. Researchers found that these baby toads take several times longer to react and move away from an approaching ant compared to native frogs. And once an ant does grab hold, the toad's famous toxin does nothing to stop it. Ants don't have the kind of circulatory or nervous system that the toad's poison is built to attack, so a swarm of meat ants can bite, hold, and overwhelm a toadlet with no real risk to themselves. In one of Team Bufo's more inventive field experiments, the researchers placed small amounts of cat food near the edges of ponds to attract higher densities of meat ants toward the shoreline where baby toads were emerging. The results showed a clear increase in ant activity and, along with it, a sharp rise in the number of toadlets being killed in those areas. Follow-up studies published in the Journal of Applied Ecology found that in high ant density zones, the vast majority of emerging toadlets were detected and attacked by ants within minutes of leaving the water, and most of those attacked did not survive. It was one of the first pieces of evidence that a native Australian species could meaningfully reduce cane toad numbers without needing to be introduced, engineered, or managed in any complicated way. The ants were already there. They just needed a reason to concentrate their hunting in the right spot. Other researchers looking at the same shoreline habitats found something almost as interesting hiding in the mud itself. Hoofprints left behind by cattle and horses create small pits along waterway edges, and young cane toads are drawn to them because the depressions stay cool and moist compared to the surrounding dirt. But those same pits can become traps. In hoofprints with steep, straight sides, toadlets struggle to climb back out, which gives patrolling meat ants far more time to find and kill them. It's a small, almost accidental example of how the landscape itself, shaped by decades of livestock grazing, has quietly been working against the cane toad's youngest and most vulnerable stage. Even with all of this, the ants alone haven't been able to stop the toad's spread across the continent. And it's important to be honest about that. Their impact depends heavily on local conditions. In some waterholes, ant density is high, the terrain funnels toadlets into a tight, exposed strip, and predation rates are dramatic. In other areas, there's more vegetation for young toads to hide in, fewer ants patrolling the shoreline, or simply more space for toadlets to spread out and avoid detection. Some toads also manage to climb onto rocks, logs, or debris where ants can't easily reach them. So while meat ants are a genuine and measurable pressure on the population, they're one layer in what has become a much larger, more coordinated strategy rather than a single miracle fix. That layered strategy has expanded well beyond ants. Water beetles and aquatic insects have been found preying on cane toad eggs and tadpoles while they're still developing in ponds, meaning the toads face threats before they even reach the vulnerable toadlet stage that the ants exploit. In parts of Western Australia's Kimberley region, researchers have documented native water rats learning, in the span of just a few years, how to flip cane toads over and eat only their hearts and livers while avoiding the toxin-laden skin and glands entirely. It's a striking example of native wildlife adapting on its own timeline to a threat that arrived less than a century ago, and it offers a genuinely hopeful sign that some Australian species are capable of learning around the toad's defenses. The most ambitious effort yet, though, comes out of a lab rather than a waterhole. A team led by Rick Shine, now at Macquarie University, has used gene editing tools to knock out a single gene in cane toad eggs, a gene responsible for triggering the hormonal changes that let a tadpole transform into an adult toad. The tadpoles that hatch from these modified eggs never make that transformation. Researchers have nicknamed them Peter Pan tadpoles, because they simply stay tadpoles, growing larger and living in the water for months rather than the few weeks a normal cane toad tadpole spends there. Because they never grow up, they can never breed and never spread the trait on their own, which limits any risk of them becoming a new problem in their own right. What makes these tadpoles genuinely useful is a behavior cane toad tadpoles already display in the wild: cannibalism. Ordinary cane toad tadpoles will eat the eggs of their own species when they come across them, but the gene-edited Peter Pan tadpoles, growing larger and staying active in the pond for far longer, have been found in trials to consume several times more eggs than their unmodified counterparts. In some experimental ponds, when older Peter Pan tadpoles were present, researchers observed close to zero survival among newly laid cane toad eggs, while native frog eggs in the same water were largely left alone. Because the toad's targeted gene is simply switched off rather than replaced with genetic material from another species, the modified tadpoles don't meet the legal definition of a genetically modified organism under Australian law, which has made the approval process for limited trial releases somewhat more straightforward. Even so, the scientists behind the project have been careful to stress that this is still an early-stage tool, tested so far only in controlled ponds, and that scaling the idea up to a continental level remains a serious technical challenge, since each modified egg currently has to be produced individually. What ties all of these threads together, from a cat food experiment beside a Northern Territory pond to a gene editing lab at a university hundreds of kilometers away, is a shift in how people think about controlling an invasive species. The original 1935 plan failed precisely because it tried to solve a problem by introducing something new and hoping it would behave predictably. Nearly a century later, the most promising strategies are doing almost the opposite. Looking closely at animals and processes that already exist in the Australian landscape, and finding the narrow moments where the cane toad, for all its toxicity and its sheer numbers, is still just a small, soft, thirsty little creature standing at the edge of the water. The cane toad invasion is far from over, and no single tool, whether it's an ant colony, a learning water rat, or a modified tadpole, is going to erase 200 million animals and a century of spread on its own. But the fact that this story now includes native predators quietly adapting, researchers finding cracks in an animal once thought untouchable, and technology being used with real caution rather than the recklessness of 1935, says something worth paying attention to. Sometimes the most powerful response to a mistake in nature isn't a bigger, bolder intervention. It's patience, close observation, and letting the ecosystem's own smallest residents do what they've quietly been capable of all along. What do you think is the most promising way forward here? The ants, the tadpoles, or something else entirely? Let us know in the comments, and if you want more stories like this from the natural world, make sure you're subscribed for what's coming next.